Welcome, folks, to Chapter 17 on immunizations. So in Chapter 16, we had talked about the adaptive immune response to pathogens. And so essentially, when there's an infection by a pathogen, the immune system still needs to learn about that pathogen, specifically as an antigen within the body, and how it can recognize it, and then how it can remember that for subsequent infections. And actually, in subsequent infections, then oftentimes the immune response is much more quick, much more effective, much less likely to result in illness or significant illness. And so with that knowledge, how can we, within artificial mechanisms, actually induce an immune response without inducing illness so that secondary infections can then be presented by the immune system's effective kind of response and then be less likely to induce illness. And that's essentially the purpose of vaccinations. So taking the kind of experimental nature out of how your immune system has to develop recipes for kind of understanding the pathogen, and then later have that memory of the pathogen, and instead just giving the immune system the recipe for recognizing that pathogen, that antigen, and then being able to subsequently respond in an effective manner. And so that's what we're going to be talking about today in Chapter 17. So I recommend you pause the video, the lecture video, review your notes from previous chapters, and also look over Chapter 17, and then come back into the lecture video. Okay, welcome back. So at the end of Chapter 16, we talked about the mechanisms through which we develop immunity. Some of them are natural and some of them are artificial. And so Chapter 17 focuses on artificial mechanisms for developing immunity. And so we'll talk about active immunizations, which is essentially vaccination, and we'll mention passive immunotherapy as well. So as we mentioned before already, we know that, and historically, folks, recognized and realized that oftentimes an initial infection or illness would then prevent subsequent illness in individuals, in children, so on and so forth. Also in the 1700s, with the work of Edward Jenner and others, there was a realization that similar viruses or pathogens could also sometimes lead to immunity. And so specifically with milkmaids that were being infected by cowpox, which was similar in nature to smallpox, and then they were actually developing immunity from smallpox as well via that similar pathogen that they were infected with. And so there was research at the time around actually using the result of infection. So, for example, the scabs from smallpox or pus from various illness to actually induce a milder version of the illness in other hosts, in other humans, and then allow for that to lead to immunity for subsequent infections. And initially, I mean, even to us, that sounds like kind of a strange idea, but as the mechanisms for what was called at that time virilation, and is now usually referred to as vaccination, have kind of developed over time, it's become a pretty common method for preventing against illness, and it's had significant just public health impacts overall. We're going to talk about some of those specific examples as well. And so here you can see the examples of the prevalence of both polio and measles within the American population. And so just over time, first, both of these being pretty common childhood diseases or illnesses, and then eventually after the development of vaccination, them becoming, in the case of polio, essentially eradicated in the United States, and in the case of measles, this vaccine has been about 90-something% effective at reducing cases of measles. Okay. It's important, we won't focus a lot on the social impacts of vaccination, but it's important to consider that there are inequities in availability of just healthcare generally throughout the nation and throughout the world, and specifically with access to vaccines. There are pathogens that we just haven't been able to develop vaccines for various reasons, because of, you know, how they infect their host, or the mechanisms by which they are spread, or maybe because of the anatomy of their, you know, antigens, so on and so forth. And so there are pathogens that still vaccines are not available for. And then just the fact that vaccines are very kind of financially and from a financial perspective, manufacturing perspective, time and energy perspective, they're very kind of resource heavy to develop, and therefore there has to be some sort of financial driver for them to actually be developed, and for that actually to occur. Okay, so let's just specify what the definition of a vaccine is, which we all have an idea of what it is. But vaccinations essentially are giving a new host an antigen or parts of an antigen, so that host can actually, their immune system can actually mount a response and develop memory of that antigen in most cases. And so ideally, a vaccine is easy to produce, it's easy to administer, and it elicits a similar immune response to the actual pathogen, but without causing disease. And vaccines can be developed through the use of pathogens themselves, relatives of pathogens, or parts of pathogens. And we're going to talk about that. And the goal with vaccinations is, one, to produce immunity in the individual themselves that's receiving the vaccination, but more so to produce what's called herd immunity. And that's when you have a certain quota of the population that's vaccinated, then that will actually inhibit the spread of that pathogen. Now, herd immunity can also come along through just natural mechanisms when a certain number or a certain proportion of the population has been infected and actually produced illness related to a pathogen. But if vaccines can circumvent that natural process and actually decrease the prevalence of that pathogen and that illness within the population, that is essentially, it prevents all of those negative side effects from individuals becoming ill, and then the society or the health system having to deal with those illnesses and medical facilities, so on and so forth, and also the subsequent long-term effects of illness. And so all of that, if that can be circumvented by vaccination instead of developing natural immunity through illness, then that would be the goal. And so depending on the pathogen, depending on the illness, vaccinations come in various types, and they have various kind of levels of efficiency. And so we're going to be talking about each of the different types of vaccinations separately. And so essentially, we'll talk about vaccines first and the different types of vaccines, from attenuated, inactivated, all of these, and then we're going to mention passive immunity. And what I'm going to recommend is you develop a table that looks something like this, or, you know, make it your own, that gives you the different categories. So what is active artificial immunity? What is passive artificial immunity? And then specify the different types of vaccines that we'll talk about in terms of live attenuated, inactivated, toxoid, recombinant, and combination. And then specify what is it, what are some of the pros and cons, what are examples of pathogens that have actually been, vaccines that have been actually developed against various pathogens for this type of vaccination, how is it manufactured, or some examples. We won't be able to go through how each one is manufactured, but some examples of manufacture, and then additional notes. This is actually a little bit more extensive than what's in your textbook, but since vaccinations are such a relevant topic, it's really important to kind of have this detail. Okay. So we're going to go ahead and start with live attenuated vaccinations. Essentially, you can kind of see these categories based on their name, right? So live attenuated is a living pathogen versus inactivated versus various components of those pathogens being utilized in the actual vaccine. Okay. So in terms of live attenuated, what you're looking at is a vaccine that's been developed based on actually growing, you know, actually, for example, fermenting an actual bacterial pathogen, right? or growing a virus, for example, and then using that pathogen within the vaccine that's being used. Now, that seems like problematic, right? Like, why would you infect folks with actual living pathogens? And what the key term here is attenuated. So what attenuated means is that pathogen has essentially been grown under conditions that decrease its actual virulence so that it looks similar to your immune system, but it does not produce illness, right? And so some of those ways are, for example, to grow a bacterial pathogen or a virus under less optimal conditions for that organism's growth, so it starts to mutate, right? But it's essential that those mutations still render that pathogen similar enough to the pathogen that generates illness in kind of the wild, that your body kind of recognizes them as similar without that virus, that attenuated virus or bacteria or pathogen, whatever it may be, actually producing illness in the person that's being vaccinated. And so the benefits of this, so live attenuated pathogens act like a pathogen, right? So they actually metabolizing, they're actually active, they're actually reproducing, and therefore they're kind of a complete antigen with all of the different kind of epitopes that the immune system might recognize, with all of the different metabolites, not exactly the same necessarily, but similar metabolites to those pathogens that are in the wild that could actually produce disease. And therefore, it gives your immune system kind of a diversity of recognition around that pathogen. And then it's going to lead to much more long-term immunity and much more effective immunity against that pathogen, because you're developing multiple antibodies, you're developing kind of multiple ways of recognizing that pathogen, rather than just looking at a piece or a particle or an inactive microorganism that's coming in through the vaccine. Sometimes live attenuated vaccinations will actually lead to a mild form of the illness that can actually be passed on to folks that are in the vicinity of the person that received the vaccine. And in that way can actually spread immunity to those folks without really causing the full-on illness or disease. And that's called contact immunity. Now, the reason why this doesn't work for all pathogens is for those same reasons, right? Because when you're actually infecting a new host or a person with a live, even though it's attenuated or a relative of that pathogen, then you could actually see illness in that host. And this is particularly an issue for immunocompromised folks or pregnant women, for example, fetuses, children, where that illness actually might be kind of severe enough that it results in some of the same side effects of actually just naturally receiving that illness from the wild. There are a couple of cases in which attenuated viruses have actually been able to revert to the wild form. Not very common, not so often, but it is a risk with attenuated viruses. And so some of the vaccinations that we use that are attenuated live viruses include the MMR combination that includes measles, mumps, and rubella, also rotavirus, smallpox, chickenpox, and then yellow fever. Polio in some cases used to be on this list, but there were actually some examples in which polio was reverting back to its wild form, in which it could actually cause disease. And so that was kind of eliminated from the list of live attenuated viruses that were accepted in the United States and other places. So in a case like that, where you actually have a pathogen that might actually revert to a wild type and become virulent again, what would you use instead? Well, you can actually produce the pathogen, the virus, or the bacteria, for example, and then inactivate it. And so these are inactivated vaccines. And you can either use the whole agent, where you're using the full pathogen, but it's dead, or you can use subunits of the pathogen by actually growing the pathogen and then kind of dividing it into its subunits, okay? And these can be used as vaccines. Now, as you can imagine, if those pathogens were alive, like in the live attenuated vaccines, then they would produce more kind of epitopes and more factors for your immune system to recognize. Whereas if those pathogens are killed, or especially if they're just subunits and they're not actually the full pathogen, then your immune system just doesn't have as much to recognize. And so the immunity based on inactivated vaccines tend to be lower overall and less long-lived than immunity that's developed through vaccines that actually have the live attenuated pathogens within them. Okay. Another thing is that since there are kind of subunits that are kind of within the vaccine, oftentimes they need to be stabilized with adjuvants. And these are chemicals that sometimes folks will actually have allergies against. And so that becomes a risk of inflammation and allergies kind of added to because of these chemical adjuvants that are present. And so some examples are the vaccines for hepatitis A, the flu influenza, which we take every year, is an inactivated vaccine. Polio, the polio that's used more commonly now, which is a shot instead of the oral one that had been used previously historically, and also rabies. Although there is some research looking at some new research around rabies, yellow fever, even the flu. We're going to talk a little bit more about that as well. So let's talk about production, okay? And the initial parts of production will be a little bit similar between our attenuated live and our inactive, but essentially we'll produce that virus or bacterial pathogen, and then we'll inactivate it if we're talking about the inactivated vaccines. And so for viruses, and this is an example with flu, often the virus, remember, is not a living microorganism, right? And so it has to have cells of a host for it to be able to actually divide and grow. And so oftentimes eggs are used, chicken eggs are used to provide host cells for that virus. Sometimes mammalian cell cultures can also be used, but it's a pretty intensive process because you have to have these facilities where you have a significant number of these chicken eggs, and it's a very labor-intensive process to open up just the top of them and then inoculate that virus into the egg. Usually it's about 1 dose that comes off of each egg, and then anyone with egg allergies becomes kind of an issue for them. And so in that case, you actually are producing the full virion. And then if it's an inactive activated vaccine, then you're using chemicals, heat, centrifugation, sometimes various methods to actually break apart that microbial agent, if it's a virus or microorganism, if it's a bacterial pathogen, into its subunits or actually kill it. And then that's what's used in the vaccine. Of course, there are still, a number of steps here that allow for purification of, for example, the egg components and various other byproducts before it's actually made into a vaccine. So this is a much simplified image here. This gives you an idea of how labor-intensive egg culturing can be for the production of specifically viral vaccines. Okay. Here's another example with the human papillomavirus, in which instead of actually culturing the whole virus, we actually use plasmids. So remember when we had talked about the use of plasmids in lab, we use plasmids to just take the genetic information for the capsid proteins. So those proteins that are on the outer surface of the pathogen. Those tend to be most relevant in terms of kind of recognition by the immune system, obviously, because that outer portion is what's actually binding with the cells of the body and then actually infecting those cells. And so oftentimes when we're talking about subunits, we're talking about subunits on the external surface of the antigen. Okay, and so in this case, we actually just take the gene for some of those proteins. Here, let's get a little bit closer from some of those capsid proteins. And we, through recombinant DNA methods, which we have used in the laboratory, in our lab, we actually grow just those proteins via some sort of other organism. Oftentimes it's yeast, and sometimes it's mammalian cancer cells. Okay. And then those subunits are purified, and those steps of purification are also labor intensive and manufacturing intensive. But then you get this purification of just those protein subunits that can then be injected or through vaccination into a new host, and then the body can actually mount an immune response against them. And so that gives us an idea of how both whole live attenuated viruses as well as subunit viruses are made. And there's different ways depending on the subunit that's being made, right? But you could be growing the whole pathogen, you could be growing parts of the pathogen. Recombinant methods are very commonly used in actually just selectively producing subunits of pathogens, for example, in what we saw with the human papillomavirus. Okay. Okay, so that's live attenuated, and then our inactivated, either whole inactivated or subunit inactivated viruses. So let's move on to toxoid viruses. Toxoid vaccines. Toxoid vaccines are specifically a really good mechanism for use against pathogens that the primary antigen is not the pathogen itself, right? So, for example, Clostridium tetani, and then current bacteria diphtheriae, both of these, the cause of illness is not necessarily the pathogen that's actually infected and growing within the organism, it's the toxin that they produce. Okay, so the tetanus toxin, which is a neurotoxin, for example, that's produced and then actually stiffens up muscles until the patient dies, for example, right? And so in these cases, since the toxin is the primary antigen, we can take that toxin and then we can inactivate the toxin rather than taking the pathogen and inactivating the pathogen. Because again, the pathogen is not the primary antigen in this case. It's actually the toxin that's being released or produced. And so we take that toxin and we inactivate it, hence the term viroid, hence the word toxoid. Toxoid being, pseudo-toxin or inactivated toxin. And that is what's administered in the vaccine, okay? And so is very effective against these specific pathogens that are actually releasing toxins. Since though, if you're looking at a toxin, right, if you think about the immune system response, you have the pathogen, the active pathogen, the way in which it produces metabolites, and then the toxin itself. And then your immune system in a live wild type infection can kind of recognize all of that and respond to all of that and remember all of that. Whereas in a toxoid vaccination, all you're giving your immune system is just that one chemical, right? That one toxoid. That's a relative of or an inactivated version of a toxin. And so it just doesn't have a lot to work with in terms of immunity and memory over time. So oftentimes toxoid vaccines, you have to have consistently boosters throughout your life to be able to kind of maintain that immunity over time. Okay, and so we had mentioned diphtheria and tetanus, and we know that both of these we basically have to get every 10 years to maintain our immunity against. Okay, in terms of production, what you're doing is literally culturing those organisms. So this is a large excuse me, a large scale fermentation process where you're actually culturing these types of Clostridium or foreign bacteria. And then you're actually then purifying off that toxin and then inactivating it, oftentimes with formaldehyde or other types of chemicals that will inactivate the toxin. Similarly, some of those same chemicals are what we use when we're talking about inactivated whole vaccines. So when we said we would produce a pathogen and then kill it, right? Some of those same chemicals are used to inactivate pathogens, formaldehyde being a very common one. And then once you've purified that toxin and inactivated it, then you can actually utilize it as a vaccine. And again, these are really labor-intensive mechanisms because you're working with live cell culture. You're working with live cell culture of pathogens, right? So lots of biological safety levels, so on and so forth. And then a number of, just a series of manufacturing steps to then effectively purify that toxin and then inactivate it and then purify it again once it's been inactivated. And before you have the development of a vaccine. And so that gives you an idea of why there's so many, monetary constraints and manufacturing constraints, which you had to mention quickly at the beginning of the lecture on the production of vaccines. And there really has to be a driving force for them to actually be produced. Okay, let's continue forth and talk about the next type of vaccine, which is the recombinant gene technology. Now, this is actually a really big category of vaccines that includes using DNA, using RNA, using different kind of mechanisms within that biological dogma, right? So from DNA replication to transcription to translation. So DNA, which is replication, DNA to RNA, which is transcription, and then RNA to protein, which is translation. And so somewhere along that line, being able to essentially give the vaccinated individual, the new host, just the code for parts of that pathogen that their own cells, their own body will then actually encode and produce, and then the immune response can be developed based on that. Now, historically, DNA vaccines had been there were a lot of research around DNA vaccines, but there's a lot of just difficulties around how do you actually how do you actually transfer that DNA into the host? Oftentimes it's through a viral vector that is delivering that, and so on and so forth. But now in the last couple of decades, 20 some years, there's a lot of research on RNA vaccines, and actually the COVID vaccine is a result of that research. So here, DNA vaccines, this is a West Nile virus. You're taking, for any recombinant vaccines, you have to know the pathogen really well. You really have to have an architectural kind of layout of exactly what the pathogen looks like, exactly all of the genes that are being encoded by that pathogen, and which of those pieces, which of those proteins or which of those components on the outside of the pathogen are actually what's kind of causing infection or allowing for infection, and therefore being able to kind of isolate, okay, it's this protein that I'm going to isolate the gene for, and it's that protein that I'm going to have the cells of my new host that's actually receiving the vaccination produce to elicit an immune response. So there's a lot of bio kind of tech and biological engineering that goes into understanding exactly which components need to be utilized in recombinant vaccines, in recombinant genetic vaccines. And so that, that understanding of the pathogen is essential. And maybe there are some pathogens in which we don't have that understanding of exactly which antigens would be utilized in a mechanism like this. Okay, so back to the West Nile virus. You kind of look at the virus, you identify some of the components of the virus. So these outer structural proteins, the green one and the blue one, that can be utilized in these, DNA recombinant vaccines. And then you take those genes specifically and you actually put them onto a plasmid, just how we had done in the PGLO we had talked about with the PGLO experiment in lab. So now you have a plasmid that actually has those genes on it, and then you use a vector to actually infect the host with those genetic pieces, and then the host themselves, as a human host, we then will then make those proteins ourselves. Our cells will actually make those proteins, and then the immune response will be developed against those proteins that the human cells themselves have made, right? And so, again, this is kind of manufacturing. All of this requires that these plasmids can be made. They also have to be cultured in living cells. Oftentimes, those are human mammalian cancer cells, for example. And so it's kind of an intensive process here. But it can be effective, although there are not very many examples of DNA vaccines. On the other hand, if you eliminate all of those initial layers, right? So instead of starting with DNA, which requires, the replication and then the transcription and then the translation, start at the end of that series of steps, right? Start with mRNA, which is literally the genetic code that's used in the last step to then generate proteins, right? And so for the last couple decades, there has been work with mRNA viruses, flu viruses, some work around HIV, but that's still kind of being developed. And so just years and years of work around mRNA viruses. But for a long time, the difficulty was, how do I get a piece of mRNA into a host when that mRNA itself is very unstable, right? And so the delivery mechanism, right? So you could use viruses for this, but that has its own kind of host of cons as well. And so recently, in the last couple of decades, so here I'm going to show you this top image right here is going to change, and then we're going to kind of go back to the same content here. So for example, with the coronavirus, the corona is a crown, which are these kind of spike proteins that are on the outside of the virus. And you can actually identify a specific genetic code that codes for just that protein. And so for a long time, we had the code, we had the mRNA, but the delivery was the mechanism. And then with developments in nanotechnology, what we could do here, this black strand of mRNA is equivalent to this tan strand right here of, kind of the copy of that genetic piece of information. With nanotechnology, we're actually able to develop lipid casings, lipid nanoparticles that would spontaneously cover the mRNA particles, the mRNA molecules, and then that allowed for easy transmission or easy, take up of those mRNAs into the new host via vaccination. And so it was essentially those developments in nanotechnology that really moved this research forward, even though it had kind of been considered for a long period of time. And so the first example of an mRNA vaccine was actually in 2020 for Ebola. And there were a significant number of doses that were given out. But essentially, since this goes back to the social kind of impacts. Since Ebola tends to be localized, although it does have the potential to become a pandemic, just wasn't enough funding, and there wasn't enough kind of resources that went into continuing to kind of mass produce and develop that mRNA virus. And then, of course, the pandemic then brought that level of interest and resources into the picture. And then we were able to pretty quickly develop on a mass scale, these mRNA viruses. And this is the first time that we've seen this mass scale of this, type of technology being used for vaccination. Mass scale of mRNA vaccines. And so this is kind of that first time. And so one of the reasons why it was so easy to kind of quickly develop and turn around these vaccines is because it's a completely artificial process, okay? And we're going to look at a flow chart of how it happens. But for the other examples of production and manufacturing, we talked about needing chicken eggs, we talked about needing mammalian cell cultures, we talked about needing large fermentation vats where you're actually literally producing the pathogen itself, right? A lot of safety kind of considerations that go into that, large scale manufacturing, labor intensive manufacturing, But if you have a completely artificial process, then you don't need all of those levels of safety. Well, how do I manufacture, large vats of a pathogen, for example? There's just a lot of safety and a lot of kind of approvals that need to go into that. But if you have a completely artificial process where you're not producing any cell lines, you're not producing any pathogens, then it allows for a much more kind of expedited approval process. Okay. And so that's really the key with the mRNA vaccines, is that they're artificial in terms of manufacturing and delivery. You're not using a virus for the delivery of the mRNA anymore. You're just using these lipid nanoparticles. On top of that, because you've eliminated all of those excess steps, specifically with cell culturing, you can get very high and clean yields. So remember, in those previous diagrams, there were all of these steps for purification, because you have all these other cell components that you had to generate, or egg components that you had to generate to get the vaccine. But in this case, you don't have those. And so it just makes it a cleaner product, and then you can have higher yields. And so you can actually get about 50 to 60 doses under certain conditions, not all conditions, of some mRNA viruses per milliliter, which is unheard of. I mean, we were previously in some of those viral vaccines that we had seen, we're talking about 1 dose per egg, right? And so just a much more efficient process overall. And then when it comes to the efficiency of the vaccine and the immune response that's developed based on the vaccine, they're pretty much similar in terms of previous mechanisms for developing vaccines. And then I think we talked about this, simpler and quicker for manufacturing. In terms of cons, you really remember what we said when we were talking earlier about the DNA examples of DNA vaccines. This is true for any type of recombinant vaccine. You absolutely have to have a very, very clear understanding of that pathogen in terms of it being an antigen that is recognized by your immune system. What are the components of that antigen that are most effective in terms of developing an effective long-term immune response? Because you have to you're just taking just the genes for those specific components. You're not producing the whole antigen. And so really understanding the pathogen is absolutely essential, and we just don't necessarily have that understanding for all potential pathogens. And then once you have that understanding, then genetically engineering, which genes that you're going to produce, and then making sure that when those genes are actually, those mRNA particles are actually translated, the protein that is produced is also folded correctly, right? Because that has to occur kind of autonomously without the cellular mechanisms and structures. And so that essentially, that component is kind of the most intensive component of producing these mRNA viruses. Specifically for mRNA viruses, depending on the virus, I keep on saying viruses instead of vaccines, but specifically for the mRNA vaccines, there are examples in which the vaccine can be used without an adjuvant. Remember, those are the chemicals that help stabilize and stabilize the antigen, but also activate the immune system. But sometimes they're still needed, and so you still have that added risk of inflammation and allergies because of those adjuvants in that case. And then, of course, since we're just starting to utilize these mechanisms on a large scale, understanding how they can be improved over time is essential as well. And so here's just a simple flow where, again, just this piece, that it's a cell-free synthesis. There's no living cultures that are needed. It's fully an enzyme-based. You have your mRNA, you have your enzymes, and that's the full extent of how that is produced without cells, right? Of course, there are other chemicals and things as well, but in relation to cells. And then purification is much easier because you have fewer inputs. And then the filtration is also, again, easier because you have fewer inputs, and then you have your final mRNA drug or vaccine, and it tends to be in large doses per milliliter. So that's also a benefit as well. More specifically related to COVID-19, you can see again here, this is the COVID-19 virus. This is a spike protein that's actually kind of interacting with the human host cells and then allowing for infection. And so looking at the genetic information and then just taking those genes that code for that spike protein and then utilizing those in the vaccine, okay? And so those are essentially vaccines of mRNA and then the enzymes that are needed to kind of accompany that. Okay, and so the mRNA enters the cell, then the cell itself, this is another key, is that the host cells are the ones that produce the antigen, right? In other types of vaccines, the vaccine is carrying the final antigen. But in mRNA vaccines or in recombinant vaccines generally, the host cell, our cells, are the ones that are actually producing the antigen, displaying that antigen, and then the immune system is then responding to that antigen that was produced, internally by those host cells. And so that's another really big key difference between mRNA vaccine technology. And then again, you have your antibodies that are produced, and then your memory cells that are developed. And then if there's a subsequent infection, then you can recognize and efficiently kind of respond to. Now, similar to toxoid vaccines, you're only producing one component or one subunit of that pathogen or that antigen, right? And so the immune response is not going to have a diversity of different types of epitopes and different types of metabolites to recognize off of that pathogen. And that's why you're going to, just like with toxoid vaccines, but even more so, and subunit vaccines, which we had talked about previously, you're going to need multiple boosters, especially if it's an organism or a microbial agent that's mutating quickly over time, okay? And so that's why we see a number of the boosters being needed against these, specifically COVID-19 or others as well. So far, we've talked about live attenuated vaccines. We've talked about inactivated vaccines, which have two sub kind of categories, which are whole killed vaccines and subunit vaccines, okay? Similar to subunit vaccines, we've talked about toxoid vaccines, which instead of taking a subunit of the pathogen itself, we actually take the toxin that's produced off of that pathogen and we utilize that in the vaccine. And then we've also now talked about recombinant vaccines. So we gave some examples of DNA recombinant, but then also just really relevant example of some of the new mRNA vaccines that are being produced. Okay. Now, as some vaccines have actually been approved in combinations where you're administering the vaccines for multiple different pathogens at once. And this is not possible for all vaccines, like just get one shot and that's it, you're done. But there are some that have been approved. So we're familiar with these DTaP combined vaccines, which are combinations for diphtheria, tetanus, and others, and then MMR, which is a combination for measles, mumps, and others. And so when you get that one vaccine, then it helps, provides immunity against multiple different pathogens and illnesses, and still oftentimes you need to get boosters over a lifetime. And so those are our types of vaccines. And so essentially, regardless of the type of vaccine, although of course there are differences in terms of the details around each, when you get the vaccine, your body responds with an immune response that allows you to develop antibodies and allows you to develop memory around that pathogen. Depending on the type of vaccine, those are going to be more or less long-term responses, right? So if you have a whole live vaccine, then you tend to have a longer kind of memory of and ability to respond against, or more effective ability to respond against subsequent pathogens or infections. Whereas if you're working with subunit or for example, subunit or toxoid vaccines, then you're going to need subsequent vaccines. But essentially, your body has those antibodies and those memory cells related to that infection and can mount a more effective subsequent response to an infection by that specific pathogen. Now, how do we know if that's occurring? We can actually test the presence of antibodies in the blood serum of the host that's received that vaccination, or even if they have had an infection. And so I wanted you to see a video of one of those mechanisms. There's many different assays that you can use, but because it relates to some of the natural kind of impacts of the immune system and antibodies, specifically related to agglutination, which we had talked about before. And so there's a video on testing for antibodies in blood serum. Go ahead and just watch that video and look at that process of how we can kind of see whether a patient is actually developing those antibodies based off of a vaccine. But this could also be used if they actually have the illness. Now, not everyone is tested for antibodies, but there are mechanisms so that we would know if patients have those. So go ahead and pause, watch that video, and then come back into the exercise. Okay. So just generally and related to vaccines, when we had talked a little bit about potential for mild infection or toxicity, and then the risk of allergic responses, particularly to adjuvants. But in terms of allegations that vaccines cause autism, diabetes, asthma, various types of neurodegenerative diseases, there is not research that substantiates this, okay? And so vaccines, they provide this immunity against, herd immunity for, and then just decreased levels of infection that allow us to circumvent a lot of the negative impacts of illness overall. And so we've, in many of these cases, polio, for example, measles, for example, we've actually even lost the memory oftentimes just in the general population, not in the medical world, of what these illnesses look like. And that's because we don't have to experience them. And that's mainly because of vaccines. And so that really circumventing all of those negative impacts is a really huge win when it comes to just medical advancements overall in terms of vaccinations. Your book provides a kind of recommendations, kind of a schedule for different types of immunizations and who should receive them over time. And so we're familiar with this as well, kind of in our daily lives, right? So I would invite you to go back to this table that you had developed earlier, or a variation of this table. Make sure that you have all of the information for the active artificial immunity kind of filled out. And then we're going to move into just a quick discussion of passive artificial immunity. Okay. Okay. So in our last video lecture, we had just introduced passive immunotherapy, which is essentially when you take antibodies that have been produced by a different host and you put them into a new host, okay? So the essential difference between passive immunotherapy and vaccinations is that the antibodies in vaccinations are produced by the host that actually receives the vaccination. Whereas in passive immunotherapy, those antibodies are produced by a different host, okay? A different human host or a different mammalian host, that then those antibodies are then given to via, serum injections into the blood via to that new host, right? And so those antibodies are not being developed by those hosts, okay? And so essentially what you can do is the passive immunotherapy becomes important in cases where a host is exposed to a very detrimental or potentially fatal toxin or disease, right? Or potentially has been exposed to. And so this allows for us to circumvent kind of the whole immune response process and then just immediately give them the antibodies that allow them to neutralize that antigen immediately, right? And so what this looks like is oftentimes we'll have mice, rabbits, horses that have been infected with some kind of similar pathogen, and then off of their blood serum, the antibodies are actually harvested and then given to a host, a new host, as an injection, an anti-serum injection. And then those antibodies within that injection, then essentially they bind to the antigen, which is the pathogen or the toxin, and then immediately they can kind of start to neutralize that pathogen. So we had talked about this in terms of snake bites and venom, also, tetanus, for example. And so these allow us to really just, Ebola is another example we had given, and this allows us to essentially neutralize that threat that we know is already present in the body of the host. Now, the really big key difference, because you might be thinking like, don't we just do this all the time? Why don't we just take the antigens and if we want to quickly prevent disease, just provide the antigens to the new host via vaccination and like skip and circumvent this whole process of, growing viruses in eggs and growing different types of pathogens in large fermentative vats and, giving them to patients and so on and so forth. Why don't we just produce the antibodies and distribute the antibodies? But remember that any foreign object in your body is going to be an antigen, even antibodies. So your body is actually going to recognize those antibodies as a foreign object and tag them as an antigen, as a threat, as an antigen. And so this is very common that folks will actually develop kind of like an allergy to those anti-serums, which is called serum sickness. And if they have to receive that anti-serum in, future, cases like future infections, then they'll actually have the serum sickness because their body has tagged those antibodies with antibodies, essentially. And so it's just not really an effective large-scale mechanism, but there are certain cases in which the threat is so high that you can use the delivery of these antibodies just to neutralize the threat. The other thing is because of, what we had discussed here, we do not get immunity against subsequent infections. If there is a subsequent infection, you still have to use antiserum again. And then we already talked about the risks with using antiserum multiple times because your body is essentially going to reject that. So passive immunity is a really, really important tool in cases where you have these imminent risks, but it's not a generally kind of a mechanism that is useful for vaccination. There's developments of these hybridomas, which are essentially human cancer cells to produce the antibodies that has reduced some of these risks, like serum sickness, because the antibodies are actually produced by a human cell instead of a different type of mammalian cell, like a horse or a rabbit or a mouse. And so that has reduced the issues with serum sickness and some of the allergies, and the body actually rejecting some of these anti-serums. That has been an issue in the past. And so that research essentially allows you to produce the antibody, but then grow it within human cancer cells so that it's less likely to be rejected. And so that research is also important to the production of anti-serums as well. So that essentially takes us to the close of our discussion of the different types of artificial active immunity that we can develop and artificial passive immunity that we can kind of develop. And so remember, go back to that table, fill that out. And what you're going to want to do for attendance is once you have that full table completed, then you just go into the Jamboard and put your name in an emoji. You don't need to post the whole table. It's just too large for that. So just, once you've completed that whole table and you're confident about it, then go ahead and jump into the Jamboard that's linked below and add your name and an emoji. If you have any questions about it, of course, as always, feel free to email me the table or any questions that you have. I would love to respond to those. Now, another couple things that are relevant to this discussion are just other technologies that are used around antibodies, and then also social implications of vaccines. So let's just have two really short discussions before we close this chapter overall. So antibodies are actually produced for other technologies as well, some of which we're unfortunately very commonly familiar with, like the COVID-19 home antigen test. And so what literally that is based off of antibodies that have been produced off of other mammalian hosts. And they're actually kind of on this paper here, they're actually held into the paper of the test. And it's interaction between those antibodies and the potential antigen that actually gives you the positive or negative results, right? And so that's a very similar mechanism for pregnancy tests as well. And so I wanted you to watch the two videos, one on the COVID-19 home testing kits and one on the pregnancy test, that gives you an idea of some other ways, some other biotechnologies that we utilize antibodies in, okay? And so go ahead and pause the video and watch those two videos, the lecture video, and watch those two videos, and then come back. Okay. So oftentimes when we start talking about vaccinations, the elephant in the room is, anti-vax controversy. And so we won't talk extensively about anti-vax controversy, but we will mention that since the days of Jenner, we've always had skepticism around vaccinations. And that makes sense, because it's literally Jenner is infecting folks with the results, or the crust or the pus of other people that had been infected with these diseases and these illnesses. And so you can already see in these images, like people growing cows because they are being infected by cowpox or whatever. There is a level of skepticism anytime there is a scientific method or a scientific procedure that folks don't understand. And so it's a combination of the research itself, the way that people kind of interpret that research, the information that they've been given about that research, and maybe larger political kind of waves or tendencies as well. And so all of these things come together sometimes, and you will have patients, many of you will be in the medical setting, that are going to be skeptical of vaccinations and so on and so forth. Even scientific research itself sometimes will actually be used to develop some of these narratives. Like, for example, correlative data that is not giving you causative relationships being used to say, oh, well, we see these types of autism or neurodegenerative diseases or so on and so forth emerging at the same time as we see vaccination. Well, that's a correlation, because those two things just tend to kind of occur around the same time, for example. So oftentimes, being clear in the way that you present these types of procedures, vaccinations, so on and so forth, and being aware of certain criticisms that folks have and being able to kind of address that is going to be a huge step towards mitigating some of these concerns and allowing for vaccination to become more widespread. And so for folks that are going into the healthcare kind of world, you guys are really going to be on the front line of this. And so understanding, not just kind of saying, oh, anti-vaxxers, and this causes a lot of issues, and we're losing herd immunity, and so on and so forth, but really starting to understand how you can clearly portray this type of information and the benefits of vaccination and the science around vaccination to folks that might be skeptical. And then you can actually be on the front lines of promoting healthcare just generally throughout your patients that you see, so on and so forth. And so there's a lot more to be said about vaccination and controversy around vaccination. And that's not our place today, but I would recommend that you really consider you being kind of that mediator oftentimes between some of these misconceptions and preparing yourself for that. The last video that we'll watch gives us some social context, not around vaccination controversy specifically, but around the implications for vaccines within the pandemic, but also within social settings. And, gives us kind of also just a review for vaccines. And so that final link that you'll see in the list of links, I would recommend that you go into that. That's also a crash course has within that is actually a series around pandemic science. And so it's a resource that you might want to look at as well. So thank you for listening. Make sure that once you finish your review of this chapter, you go into the Jamboard so that you can get your attendance for today, and you just post your name and your emoji indicating that yes, indeed, you have completed the table and the notes for this lecture. Thank you for listening, and I'll see you in the next resources for the class.